What if medicine could be programmed the same way we program software? Instead of discovering a completely new drug for every disease, scientists could simply change a genetic instruction and allow the body to manufacture the therapeutic molecule itself. This is the core concept behind mRNA therapeutics, a rapidly advancing platform that is transforming modern biomedical science and drug development. To understand why this technology is so powerful, we first need to look at the central dogma of molecular biology, the fundamental process by which genetic information flows inside cells. In every living cell, DNA stores genetic instructions. These instructions are transcribed into messenger RNA, mRNA, which then travels to ribosomes where proteins are synthesized. Proteins perform most biological functions, including catalyzing biochemical reactions, regulating metabolism, transmitting signals, and defending against pathogens. Many human diseases arise from defective, missing, or insufficient proteins. Traditional pharmaceuticals usually attempt to correct these problems indirectly, often by blocking enzymes or modifying signaling pathways with small molecules or antibodies. However, these approaches can be limited because they do not address the underlying protein deficiency itself. mRNA therapeutics offer a fundamentally different strategy. Instead of delivering a drug that modifies biological pathways, synthetic mRNA delivers the genetic blueprint for a therapeutic protein directly into cells. Once inside the cytoplasm, the mRNA is translated by ribosomes into the encoded protein. The patient's own cells therefore act as temporary bioreactors, producing the therapeutic molecule in situ. One of the most widely known applications of this technology emerged during the COVID-19 pandemic with the rapid development of mRNA vaccines by companies such as Pfizer and Moderna. These vaccines contain lipid nanoparticle encapsulated mRNA encoding the SARS-CoV-2 spike protein. After administration, host cells transiently produce this viral antigen, which stimulates both humoral and cellular immune responses. The success of these vaccines demonstrated the clinical feasibility, scalability, and safety profile of mRNA delivery platforms. Beyond vaccines, mRNA therapeutics have broad potential in several major areas of medicine. One important application is protein replacement therapy. Many genetic disorders occur because a specific enzyme or structural protein is absent or dysfunctional. Conditions such as metabolic enzyme deficiencies, for example, could theoretically be treated by delivering mRNA encoding the missing protein. Unlike permanent gene editing technologies, mRNA does not integrate into the genome. Instead, it provides transient expression which reduces long-term genomic risks. Another promising area is oncology. Cancer cells often produce mutated proteins known as neoantigens that are not present in normal tissues. Scientists can sequence a patient's tumor genome and identify these neoantigens. Personalized mRNA vaccines can then be designed to encode these tumor-specific antigens, stimulating cytotoxic T-cell responses against malignant cells. This strategy is currently being investigated in clinical trials for melanoma, pancreatic cancer, and other malignancies. mRNA therapeutics are also being explored for cardiovascular disease and tissue regeneration. Experimental studies have shown that delivering mRNA encoding growth factors such as vascular endothelial growth factor (VEGF) can stimulate angiogenesis and improve blood vessel formation in ischemic tissues. This could potentially aid recovery after myocardial infarction or peripheral vascular disease. From a pharmaceutical development perspective, the major advantage of mRNA lies in its platform-based design. Once the delivery system, typically lipid nanoparticles, is optimized, different therapies can be produced simply by altering the nucleotide sequence of the mRNA. This significantly accelerates the drug development process compared with conventional small molecule or biologic therapeutics. However, the technology also presents scientific challenges. mRNA molecules are inherently unstable and susceptible to degradation by ribonucleases. To overcome this, researchers incorporate modified nucleosides such as pseudouridine and encapsulate mRNA in lipid nanoparticles to protect it during systemic delivery. These formulations also facilitate cellular uptake through endocytosis and promote efficient cytoplasmic release. Another important consideration is immune activation. Unmodified RNA can trigger innate immune sensors such as toll-like receptors. Advances in molecular engineering have allowed scientists to design mRNA sequences that maintain translational efficiency while minimizing unintended immune responses. Despite these challenges, the therapeutic potential of mRNA is extraordinary.